Hello, everyone. Thank you for joining Fragile Nucleosome Mini Symposium. So we will have 4 speakers today. The first speaker is Reuben Franklin, who is a postdoc at UC Riverside, where he has also performed his PhD studies. Reuben obtained his undergrad degree in biology at California State University before joining UC Riverside, obtaining a PhD degree in cell, molecular, and developmental biology since 2018. He's very active in academic research mentoring and also active outside the lab as a volunteer, such as living assistant aide, court-appointed special advocate for fostering children, and emergency department volunteer. And I think this volunteering experience really say a lot about him as a very kind person. And he has also won numerous awards, such as the Transcend Postdoc Trainingship, as well as Graduate Research Mentorship Fellowship, and a Graduate Academic Integrated Community. And he has received many honors, including for his community outreach activities, as well as some travel grants for the conferences. And during his PhD, he has gained multiple extensive experience in molecular and cellular biology. In particular, today we will hear about his thesis research that focuses on histone chaperones, CAF-1 and SPT6 in maintaining divergent chromatin environments to preserve cellular identity. Welcome, Reuben.
Great, thank you for the kind introduction. And so yeah, histone chaperones, CAF-1 and SPT6 maintain divergent chromatin environments to preserve cellular identity. And so thanks for joining my talk. So that's okay. So our big question, right, is how does chromatin regulation affect cellular identity? So we have this, well-known central dogma process of replication, and then the DNA is transcribed, translated into protein, and then proteins can carry out cellular cell type specific functions. And so we're primarily interested in kind of how does this relate back to this? And so one important chromatin regulator is the histone chaperone CAF-1. And so histone chaperones in general bind histones, which are the core like particles of nucleosomes. And they can usually, at least in vitro, help kind of form nucleosomes. And so that's how you really define a histone chaperone. CAF-1 in specific maintains the nucleosome balance after replication. So when the genome is duplicated, CAF-1 actually deposits the new histones to sort of maintain the nucleosome density after the chromatin is replicated. And indeed, CAF-1 has been shown to be kind of generally very important for cellular identity. And so in many models, such as when you try to trans-differentiate or reprogram a somatic cell into an alternate lineage, so like a pre-B to a macrophage or a fibroblast to a neuron, CAF-1 actually restricts that change. And so when you remove CAF-1, it increases that efficiency. And the same thing goes for like a somatic reprogramming from a differentiated cell into a more pluripotent stem cell. And so in general, it's a protector of cell identity. And so we took this CAF-1 and we wanted to ask, like, why is it important in terms of a specific, like a myeloid progenitor? So this is a lineage committed cell. And it's more like a homeostatic state. And so how does CAF-1 sort of protect this specific cell state? And so to do that, we used this in vitro model of acute myeloid leukemia. So in normal bone marrow, you have your red blood cells and then these neutrophils that are kind of predominated throughout. But in acute myeloid leukemia, it's really defined by this sort of overaccumulation of these stunted myeloid progenitors. So these cells that don't differentiate and they just continue to proliferate indefinitely. And over 50% of AMLs actually overexpress HOXA9. So in the cell, in the culture dish, if we take bone marrow cells and just transduce them with HOXA9, what we kind of end up with is actually these immortalized myeloid progenitors. So we call them immortalized granulocyte macrophage progenitors. But these are basically what will form neutrophils. And that's what we see if we deactivate HOXA9, the myeloid progenitors will turn into neutrophils. And so using this cell model, we asked how does CAF1 maintain its cell identity, because we know it's to be a general protector. And so indeed, we did find that it kind of maintains this myeloid progenitor identity through a similar mechanism where it's promoting these chromatin barriers. So preventing kind of competing TFs or preventing any sort of off-target TF binding. And so that you have sort of the true binding sites of the target lineage only bound, and then CAF1 prevents any off-target binding. So when you lose CAF1, you actually get these kind of ectopic binding of TFs. And we call that resulted in like a mixed lineage state, which was sort of new, where we started to see, you know, this multiple multiple different cell lineage markers being expressed in the same cells. And so that's what we found before, actually. And so we kind of got us interested in how histone chaperones regulate myeloid progenitors in general. So we broadened the question to all histone chaperones. And to answer that question, we again used our immortalized myeloid progenitors, but we used an shRNA screening system to target 25 different histone chaperones. And then we looked for these shRNAs that induce differentiation into neutrophils. And so by quantifying the CD11B neutrophil differentiation marker, we kind of scored all these shRNAs. And what we really noticed was this hit SPT6 that stood out. And so, and it stood out even more so than CHAF1B. And the second thing we really noticed is that all the targeted basically histone chaperones had a higher differentiation score than our control shRNA. So that one highlights the importance of histone chaperones and chromatin maintenance in maintaining this progenitor identity. And two, really shows how SPT6 is important for this being the top hit. And so we saw before the CAF1 kind of maintenance of this myeloid progenitor. And now we have SPT6, which provided an interesting juxtaposition because CAF1, again, kind of deposits these histones after replication to maintain nucleosome balance. But SPT6, on the other hand, is transcriptionally coupled. So when RNA pol 2 is transcribing, you can potentially lose the nucleosomes, right? And so SPT6 helps stabilize nucleosomes during transcription. And so we now have these kind of two central dogma processes, so replication coupled and transcription coupled histone chaperone pathways. And so we know CAF1 is important for cell identity, and if we look at the literature for SPT6, it's also been shown in a few cellular paradigms to actually promote cell changes. So in this case, if you want to differentiate muscle cells or epidermal cells, SPT6 is important for that change. However, similar to CAF1, SPT6 also protected the pluripotent cell identity. And so we have these two central dogma processes with histone chaperones. We know they're important for cell identity, but we've never actually directly compared them in identical cellular contexts, which can be kind of provide really important context in terms of how these separate pathways are coordinated to maintain one common identity. And so we address this question using our immortalized myeloid progenitors. And we established this dual inducible system where we can use the IPDG inducer to knock down CAF1 or the DOX inducer to knock down SPT6 selectively. And so you can see here that SPT6 off results in depletion of SPT6 and CAF1 off results in depletion of CHAF1B, which is a CAF1 subunit. Sorry. And so the first thing we looked for in this dual inducible system is differentiation markers. So our myeloid progenitors, they normally express c-KIT. This is, a cell receptor. And when they differentiate, c-KIT gets downregulated and they start expressing a lysozyme gene. So we have this marked with the GFP reporter. And so you can see in GMP, c-KIT positive and GFP negative. And then neutrophils, they become GFP positive and c-KIT negative into this box. This is quantified with flow cytometry. And so as before, we saw that CAF1 did express GFP differentiation marker, but maintained expression of this progenitor marker, kind of this, so this, co-expression of these alternate cell type genes. Meanwhile, SB6 expressed GFP, and I had the concatenate reduction in the c-KIT receptor. So kind of suggesting it was even maybe a more neutrophil-like differentiation pathway. But to really sort of compare similarities and differences between CAF-1 and SB6 and then also, is it really a neutrophil? We did RNA-seq to define the transcriptional signatures of these cells. And then we compared the differentially expressed genes between SB6 and CAF-1 to see, what's the common and what's the unique transcript regulation going on. And so here, we do see actually a lot of like unique genes that are specific to CAF-1 off depletion, and then also specific to SB6 depletion. But interestingly, we also found, a large kind of percentage of commonly regulated differentially expressed genes. So these are genes that are both upregulated, whether you deplete CAF-1 or SB6. And we also noticed there's a kind of a large absence of SB6 downregulated genes. So even though most everything, so if you look above this line here, most everything that is regulated in CAF-1 off is upregulated. So there are very few downregulated genes. But if we then cross compare with the SB6 off upregulated genes, there's very little downregulated SB6 genes that are upregulated in CAF-1 off. So this is establishing one, like they have kind of this unique effect, but there is this commonly regulated pathway. But if we compare these transcriptional signatures to neutrophils, we really also establish the uniqueness of each condition. And so here's the normal like neutrophil regulated genes, so the ones that go up and the ones that go down. And so you can see SB6 off has a lot of up and down regulated genes, so this dynamic regulation, while CAF-1 off, like we said, is mostly up regulated, so we see all red. But if you look across, there's not a lot of regions that are consistent. So there's very unique effects with SB6 and CAF-1 depletion. And so to see what's really driving these effects, we performed ATAC-seq. And this is a way to really quantify accessibility of the genome or the chromatin accessibility. And then we looked at regions that are opened or closed after CAF-1 or SB6 depletion. And here, even more so than RNA-seq, we saw really unique changes. So you can see that in the corner quadrants, there's pretty much nothing that's shared. Everything that goes up seems to be uniquely in CAF-1 off or SB6 off, and same for downregulated genes. And then as another note, though, here we also see that CAF-1 off has a lot of like general opening instead of closing. So that's the sort of this trend we're seeing with the CAF-1 depletion, while SB6 is becoming more dynamic. But here we really established these unique chromatin effects, probably highlighting their different transcription and replication associated histone chaperone pathways. And so we then describe like we see these completely different regions being affected. And so we're just going to focus on the open regions for now. And we say what really defines these regions. And so we'll focus on the CAF-1 opened regions. So these are regions that increase in accessibility after CAF-1 depletion. We saw that genomically, they're pretty distributed between like genomic regions, so promoters, UTRs, or intergenic regions. And then if we look at like the chromatin marking, what we really notice is that these opened regions in CAF-1 off are uniquely enriched for H3K27 trimethylation. And so this is the histone, silencing histone mark, usually used to sort of silence these alternate lineage genes. And so I want to sum up sort of this phenotype we see with CAF-1 off. So these are genes that really respond to CAF-1 off. And we notice here, right, we have the K27 trimethyl enrichment. When we lose CAF-1, we see the increase in accessibility at these K27 trimethyl regions. And this is associated with upregulation of these genes, these nearby genes. And another thing to note here is that this is really upregulation of basically silent genes. And so you can see even in the HOXA9 off or the neutrophil differentiation condition, the genes stay silent. So these are very like a unique effect on silent genes after CAF-1 off. And so we'll turn back to the SPT6 opened regions now, because what we saw here was this really specific effect on promoters. So you can see over 60% of the open regions were at the promoter sites. And so these, again, like, the transcription start sites. And so we looked at the chromatin environment of these sites. And consistently, we saw the H3K4 trimethyl promoter histone mark, which was unique to SPT6 off and not found in the other open conditions. But interestingly, we also saw that these seem to be active promoters, so kind of active gene transcription. And so we asked, how does the promoter opening affect the gene transcription under their control? And to do that, we have the attack-seq opening or closing on the x-axis, and then the RNA-seq up or down regulation on the y-axis. And in red, we look at all the promoters. So you can kind of see in CAF-1 and HOXA9 off, there's not a lot of promoters that open. And if they do change, they seem to be closing. But in SPT6 off, we see, yes, lots of promoters are opening. And this does result in sort of a dynamic kind of up or down regulation. But if you look up here, you can see there's sort of a bias towards that up-regulated region, maybe, losing that kind of control and just sort of transcribing relentlessly, say. And so to show kind of what we see, right, we have this K4 trimethyl and acetyl co-marked region, so like active promoters. These promoters increase in accessibility after SPT6 depletion. And then this is associated with the upregulation of those genes. And so, this is so far just a trend. We see this promoter opening, we see those genes are upregulated, but we want to know what's really driving that regulation. Is it only transcription accessibility, but something might be binding there? So to start addressing this question, we looked at these genes specifically, so the ones with open promoters and upregulated by RNA level. And we took those genes and did gene ontology analysis. And what we noticed that really struck out was that in SPT6 off, the TNF alpha signaling via NF-kappa B pathway was uniquely enriched. And so this is a pathway very important for inflammation, proliferation, and also differentiation, especially in the blood lineages. So it seemed very relevant to look at it further and to really identify like, the chromatin regulation. What we can do is look for attack-seq footprints. And so using the Tobias algorithm, essentially it looks at open regions, and then it looks for these dips over transcription factor binding sites. And these dips are indicative of like transcription factor binding. So when you have a transcription factor bound to this site, it's going to decrease its accessibility a little bit. And so that's what Tobias is looking for. And so we looked genome-wide and then highlighted the NF-kappa B transcription factor binding sites. And since we see these really unique chromatin effects, we compared SPT6 and CAF-1 off to see what's really driving the SPT6 phenotype. And we noticed these FOS and Jun factors became really like highly enriched. So in this pathway, you can see they're pretty much all of the top genes. And so they have this, very, I think kind of well characterized now binding motif. And if we look at that in the SPT6 off, we can see that it is, this is what we see is it was a deeper valley, so kind of more binding, perhaps indicating more activity. And so we functionally tested this effect. And to do that, we targeted all the members of that FOS and Jun family with shRNAs. And then we co-depleted either SPT6 or CAF-1 off, and then also examined it in the normal neutrophils. And the standout candidate was this fossil2 gene. And so we saw that targeting fossil2 seemed to uniquely rescue the SPT6 off condition. And so that's what we can see here. You can see in SPT6 off, you have the SH control where GFP is induced. And then when we co-deplete fossil2, it reduces that differentiation or kind of rescues the phenotype, indicating fossil2 really driving differentiation after SPT6 depletion. However, for CAF-1 off or normal neutrophil differentiation, that reduction is not really apparent, showing kind of the specificity of the fossil2. And so we can see here, right, we have the depletion of SPT6, and then fossil 2 is depleted. And so normally where it's induced, we now see no induction after loss of SPT6. And so this really is establishing fossil 2 as this sort of driver of differentiation after SPT6 depletion, perhaps due to those open promoters. And so this is what we see kind of in this myeloid progenitor system, right? We looked at CAF-1 replication coupled deposition and compared it to the SPT6 transcription coupled histone chaperone pathway. And so CAF-1, we saw a lot more of a general effect, right? We saw general opening and upregulation of genes. And this had a really strong effect on what were normally like silent genes. And then we also see that the differentiation and the upregulation of the cellular lineage markers is driven by ectopic transcription factor activity or binding, right? So it's binding to sites. Transcription factors are binding to sites they shouldn't bind normally. And then with the SPT6 depletion, we saw a lot more dynamic gene regulation and opening of these active promoters. And so this opening of these promoters, I think the fact that they're active is why they're opening, right? And so SPT6 is probably important for really stabilizing that activity. And then when you lose SPT6, you get this kind of dysregulation where the RNA pol 2 now can just basically elongate without the proper controls in place. And we think that the differentiation is driven actually by this increased transcription factor activity at these promoters, right? So you lost that regulation, and now you have more binding of, say, fossil 2, and that's driving these myeloid differentiation genes. And I think that really sums up kind of, I think, these differences that we see with the CAF-1 and SPT6. And so with that, I want to say thank you to everybody for joining. Thank you to the Fragile Nucleosome organizers. I appreciate this opportunity. Our funders, we have NIH funding, and then as mentioned, my CERM Transcend postdoctoral fellowship from CERM. My lab and lab mates have been very helpful, as long as our close collaborators at the MERN lab and our many collaborators at various institutions that have provided lots of feedback and guidance on this project. And you can see here a ski picture of, our main lab mates. And so I guess with that, I can take any questions.
Thank you, Reuben. I think Ben has a question for the other questions, please either raise your hand or post your questions in Q&A box.
Mm-hmm. Thanks, Reuben. That was a fantastic talk, really interesting data. I was wondering for SPT6, you kind of focused on its role in transcription, but in yeast, it's been shown to have a role in DNA replication. So Catherine Miller and Fred Winston have a paper showing a role for MCM loading. And I was wondering if you thought about a potential replication basis for the SPT6 knockdown phenotypes that you're seeing.
Yes, so that's a really interesting finding that they had. And so to sort of complement that, we do have data that shows when you deplete SPT6, you actually need cell division to get this differentiation, indicating that like replication is kind of required to induce these genes as well. And so that I think that might be connected to this SPT6 role in replication, at least like replication initiation, right?
Yeah, great. Very interesting.
And then we also do see, a strong G1 arrest after SPT6 depletion.
Okay, thank you.
So you have two questions in Q&A boxes. One of the question focusing on how the Jun and the FOS are emitted early genes with very special characteristics. Can you understand why SPT6 knockdown induces this gene precisely?
I didn't see that question. Oh, in the
chat. Yeah, it's in the chat.
I see. Oh, okay, it's the second one, sorry.
Yeah.
Jun and FOS, could you understand why SPT? So there's maybe a few reasons. So SB6 is involved in transcription. And so if these genes, like you said, they're immediate early genes, so you can kind of imagine that they're maybe primed to express. And so maybe that's at the transcriptional level, maybe it's at the even post-transcriptional level. But with SB6 loss, we start to think that it's really the prime genes, these genes that have that kind of active K27 acetyl. And so SB6 is probably restricting them. And then when you lose it, they're able to respond basically.
So yeah.
But yeah, we don't have direct evidence yet to know exactly why.
Okay. So another question, it's focusing on whether there are any global changes in the levels of histones within chromatin when CAF-1 and or SPT6 are knocked down. Is there any indication that there is an increased deposition of variant histones?
Yeah, so at the protein level, we don't really see any global changes. And that might be just due to the sensitivity of our assay, I guess. But we don't see any really global loss of histones in either CAF-1 or SB6 depletion. And I don't think we've tested, and we haven't really seen any global differences in the histone variant deposition as well. But we haven't tested, with the ChIP-seq yet. So we might see more local changes. But to that note, with the RNA data, we do see upregulation of variant histones. So with CAF-1 off, we see like upregulation of the H3F3B gene. And then with SB6 off, we actually see upregulation of what would be more like neutrophil specific variant. So these are variants that normally get upregulated in SB6 depletion. I mean, neutrophil differentiation.
All right, thank you for the wonderful presentation and also answering the questions. If you have more questions for Reuben, please feel free to type it in the Q&A box. And for the time issue, we are going to move on to the second speaker.